Welcome dear learners. In this video nanomaterial second, we will discuss some important methods of synthesis of nanomaterials. Those are included in the syllabus of BSc graduate level. These methods include high energy ball milling methods, physical vapor deposition method, chemical vapor deposition method, microemulsion, and synthesis using microorganism and plant extract. And the last part of this video contain some characterization techniques of nanomaterials such as scanning electron microscope and transmission electron microscope. Now let us start with methods of synthesis of nanomaterials. There are two methods, or we can say the approaches for the synthesis of nanomaterial: bottom-up approach and top-down approach. In bottom-up approach, the particles are assembled. That means atoms come together to form clusters, and clusters form nanomaterial. In top-down approach, the particles are disassembled. That means the bulk material is converted into powder, and this powder is further converted into nanomaterials. So these are the two basic approach in the synthesis of nanomaterial, that is bottom-up approach and top-down approach. Now this tree diagram helps us how to classify how different methods are classified according to top-down and bottom-up approach. So top-down approach mainly include physical methods such as mechanical ball milling method, physical vapor deposition methods, lithography, pyrolysis, and thermal evaporations. While in bottom-up approach, we have chemical methods and biological methods. Chemical methods mainly include soil gel methods, chemical vapor deposition methods, precipitation methods, microemulsion, hydrothermal, sonochemical methods, and microwave methods. While in biological methods, nanomaterial can be synthesized by using plant extracts, some enzymes, using agricultural waste, using algae, bacterial, fungus, and actinomycetes. Let us start with the methods of synthesis. First methods of synthesis, that is high energy ball milling method. This is basically a top-down approach. That means in these methods, a bulk material is provided, and with the help of some mechanical force, mechanical grinding, we convert this bulk material into fine powder, which further converted into nanomaterial. So this method include a mechanical energy. Now working, we have different mills of these uh, high energy ball milling methods. We have different mills such as planetary, rotatory, vibratory, rod, or tumbler type. And all these mills, they have a hard steel or tungsten ball as an important component. Also, due to the mechanical energy, that means due to the friction between these hard steel balls, there is increase in temperature inside the reactor. Hence, it is always necessary to have some cryo cooling arrangement to maintain the temperature inside the reactor. And the basic part is the bulk material is grind between the rotatory balls. What are the advantages? It is useful for large scale production of nanomaterial of metal, alloys, oxides, ceramics, and composites. Nano powder of the size 2 to 20 nanometer can be produced. It is inexpensive and easy process. And what are the disadvantages? As the process involves mechanical grinding, the shape of the nanomaterial produced is irregular, and this method produces some crystal defects due to the mechanical grinding. Now this is a simple schematic diagram for high energy ball milling methods. It includes a reactor inside which we have hard steel balls, and at the center we have a rod. With the help of this rod, the hard steel balls are rotated, and a powder, that is the bulk material, is mixed with this hard steel ball, and due to the rotation there is a grinding of this bulk material between this hard steel ball takes place. and it is provided with this cryo-cooling arrangement to cool the reactor. This is another design of this reactor in which it is a spherical shape, therefore it is also called as a rotatory ball milling methods. Second methods of synthesis of nanomaterial includes physical vapor deposition method. This is basically a bottom-up approach because in these methods, uh, atoms they are arranged into the clusters and clusters further converted into the nanomaterials. Working in this technique, material that is the bulk material is first evaporated in a vacuum, and vacuum particle and inert gas form a cluster which travels towards the cold surface and results into the deposition of nanomaterial. What is the advantages? Advantages of this method includes particle size can be controlled by adjusting the pressure of inlet gas, rate of evaporations, and the distance between source of evaporation and central cold rod. What are the disadvantages? This method is not useful for high boiling material. Now this is the simple schematic diagram of this physical vapor deposition reactor, in which at the bottom we have vacuum pump as well as gas inlet, and here we have a crucible and a heating arrangement is provided to this crucible. With the help of heating, this bulk material is converted into a vapor phase, and this inlet gas mix with these vapors of the bulk material and they form a homogeneous clusters. These clusters move upward towards this N2 cooled rod, and over the surface of this N2 cooled rod, this deposition of bulk material takes place. Deposition of bulk material into nanomaterial takes place, and during this deposition, gas get liberated, and these deposited nanomaterials they get removed gradually with the help of scraper, and it is connected into the funnel at the bottom. So this is the basic working and design of these physical vapor deposition methods. Third method includes chemical vapor deposition method, that is also called as CVD. This is again a bottom-up approach. In this technique, a material is evaporated in vacuum and then it is exposed to some volatile precursor. Precursor means a reactant, which reacts with each other and form a desired product in vapor phase. And this vapor phase of the product, along with inert gas, they form a cluster and which travels towards the cold surface and results into the deposition of nanomaterial of desired products. So it is quite similar to this chemical vapor deposition method. Only the difference is that in chemical vapor deposition methods, a reaction between different precursors takes place which results into the formation of desired product, and then that desired product is converted into nanomaterial. While in physical vapor deposition method, we are directly provided with the material and only the evaporation of that material into the vacuum leads to the formation of nanomaterial. So this is the difference between physical vapor deposition methods and chemical vapor deposition methods. Advantages of chemical vapor deposition method is it is used to prepare high purity solid material. CVD is used to prepare nanofilm and nanocoating. Along with advantages, some disadvantages are also there. In disadvantages, the temperature of the reaction and the pressure inside the reactor should be carefully adjusted, otherwise it will leads to the formation of some impurities, or we can call it as a undesired chemical reactions. Now this is a schematic diagram used to denote this chemical vapor deposition method. First component is the carrier gas. Carrier gas carry this reagent along itself into the reaction chamber, or we can call it as heating chamber. In this heating chamber, sample is placed. This sample or the precursor reacts with the reagent and form a product in this heating chamber. And due to the carrier gas, product form a cluster, and the cluster of product move towards this N2 or the rotatory cold cylinder, and over this cold cylinder nanomaterials get deposited, and these deposited nanomaterials can be removed gradually with the help of scraper, and it is collected in a funnel. So this is the simple working of this chemical vapor deposition method. Next method includes microimmersion. In microimmersion, we know that immersion is a colloidal system, and it is a mixture of two immiscible liquid. If the droplets size of the dispersed phase is in nano, then it is called as microimmersion. Now, in the methods, in these microemulsion methods, an important part is the selection of surfactant. Suitable surfactants such as fatty acid or its salts can be used. One part of surfactant is mixed with one part of oil and two part of water, and a sample are mixed gradually. The mixing results into the formation of microemulsion that is called as micellar solution. What are the advantages? Advantage of this method is microemulsions are biodegradable and biocompatible, and hence it is non-polluting, and there is no any major disadvantage associated with these microemulsion methods. Next method is the synthesis using different microorganisms. Microorganisms such as bacteria, fungus, and yeast are used in the synthesis of nanomaterial. The various uh, species of these microorganisms are given, and these are the corresponding nanomaterials produced by each and every microorganism species. Another methods include synthesis using plant extract. Extract of geranium plant is successfully used for the synthesis of nanomaterial of gold, and recently many methods have been developed by the researcher for the nanomaterial synthesis from plant extract. Now the next part include is uh, the characterization of nanomaterial. The characterization of nanomaterial is done with the help of electron microscope. Two electron microscopy techniques are there, same and same, that is called as scanning electron microscope and transmission electron microscope. So let us start with the first, same, that is scanning electron microscope. This is a schematic diagram of this schematic electron microscope instrument, which consists of an electron beam, that is a source of electron, and this electron beam is adjusted, it becomes narrow, or it is adjusted due to the presence of magnetic condenser and objective lenses, and this electron is directly exposed to the specimen, which is kept at the bottom of this instrument column, and when electron get exposed to this sample, then it gets scattered, back scattering, as well as it produces some secondary electron. So all these scattered electron and back scattered electron, they get collected, and these collected electron used to construct the image of this nanomaterial. Okay, so let us discuss instrumentation. Electron beam scan over the surface of the sample. Powder sample is taken for the analysis. Specimen sample is placed at the bottom of the instrument column. Image is a surface projection of the sample. Image is constructed with the help of scattered and secondary electron produced during the scan, and it produces three-dimensional image of the sample. What are the advantages? It provides useful morphological information of the sample. Same is also used to find out composition of the sample by using techniques called as energy dispersive analysis of X-ray, that is EDAX. What are the advantages of same? Not useful for non-conducting samples such as biological samples, and it is not useful for polymeric sample due to their degradations. Now a second technique, that is called as transmission electron microscope, or TEM. This is schematic diagram of this instrumentation of this TEM. Again, it consists of the electron source, which produce a beam of electron. It is adjusted due to the presence of condenser lens. Sample is placed at the middle of the instrument column, and the electron, they get transmitted throughout the sample. A thin film of the sample is used, and electron, they get transmitted through this thin film. All these transmitted electrons, they get collected by using objective lenses, and all these transmitted electrons used to construct the image by TEM. Okay, so this is the instrumentation. Electron beam pass through the thin film of the sample. A specially made thin film is used for the analysis. Specimen sample is placed at the middle of the instrument column. Image is a two-dimensional projection of the sample. Image is constructed with the help of primary transmitted electron, and it produces two-dimensional image. Now, what are the advantages? High resolution image is obtained in TEM. TEM give external image as well as diffraction pattern, which helps to study the crystal structure. What are the advantages? It is also useful for biological sample. In this technique, biological sample is coated by the carbon film and then used for analysis. There are some disadvantages also associated with TEM. It requires large sample preparation time. This technique is expensive and it gives black and white image. So this is a schematic diagram, a colorful schematic diagram for same instrumentation. This is the electron source. Okay, these are the condenser lenses, objective lenses. Okay, and then this is a specimen sample at the bottom. Okay, it produces scattered and backscattered. Okay, this is the backscattered. Secondary electrons, backscattered electron, they get collected and used to construct the image. This is the actual image of this instrument, SEM and TEM. This is the image obtained with the help of this SEM and TEM. This is the image of nanomaterial, spherical shape nanomaterial. This is a rod shape nanomaterial, again spherical shape nanomaterial. See, this is the region. This distance is 200 nanometer, and in between this 200 nanometer, 1, 2. Okay, so 2 nanoparticles are there. That means if you calculate the size, it becomes less than 100 nanometer for each particle. So in this way, the particle size is measured in SEM or TEM. Now this slide gives us a clear-cut idea between the differences in SEM and TEM. In SEM, electron beam scan over the surface of the sample, while in TEM, electron beam pass through the thin film of the sample. Powder sample is taken for the analysis, while in TEM, specially made thin film is used for analysis. Specimen sample is placed at the bottom of the instrument column in SEM, while in TEM, specimen sample is placed at the middle of the instrument column. Image is a surface projection of the sample, while in TEM, image is a two-dimensional projection of the sample. In SEM, image is constructed with the help of scatter and secondary electron produced during the scan, while in TEM, image is constructed with the help of primary transmitted electron. SEM produce three-dimensional image, while TEM produce two-dimensional image. So this is about SEM and TEM characterization techniques. So we will stop here. Thank you.